Okay. Hi. So we're a little bit less people, but it's okay. There are some people outside. Well, anyway, did you have? I know that there were problems like with the Moodle and stuff like that, and now everything's supposed to work and be open. So please check that you can enter the Moodle and see the material that already there. You have the presentations from last class, and you have questions for the chapter. You don't have to answer them for this class. It will be for the next one. And you have a lot of material in general that you can refer to. I'm not putting everything on there. You have also all the slides I put in PDF format from last year, so you can look at them if you want to have spoilers. But in general, it's highly recommended to use the class course. And like I told you, this course goes through an unbelievable amount of information. Okay, and this is not. There's no way, like, to really get to gain insights from this course without going over the material. So again, this is your choice. Obviously, you're adults. But again, I think it's nicer when you understand. So, do you have any questions regarding like last class? Yeah, no. Yeah.
So the questions are just for us to answer?
No. The questions, we're not going over them now. We'll go over them in next class because until I finish a chapter, then we have now. So in this class, we're going to finish the first introductory chapter, and then we'll have in the next meeting we'll go over the questions that are for the relevant chapter. And most of the time, a lot of the times, I'll try to like finish a chapter in the end of the class, but sometimes it doesn't work like that. So it's just if we finish the chapter in class, so for the next class, you'll have to answer the questions regarding to that chapter. Okay? So I'll just go over very, very quickly what we went through in last class. So we talked about atomic bonds and molecular interactions. In general, we showed in the end that all these types of different atomic interactions can contribute to specificity of protein interactions and recognitions between two proteins. We talked about the concept of chemical equilibrium, and especially like the dissociation constant and equilibrium constant, and relate that to a concept of pH and buffer. And we almost finished talking about the chemical building blocks of the cells, and we only have like one more building block to talk about. And today we're going to talk a little bit about biochemical energies, mostly in the form of ATP, like the molecules that you have here. And then we're going to go to the next chapter and start going into more detail. And next chapter will be about proteins in general, like their structure and function, and so on. So, last time, we more or less finished in the chemical building blocks of the cell. We talked about the fact that we have amino acids, nucleotides, monosaccharides, and lipids, which will be the last one. And each one of them has their polymeric or poly form. For amino acids, they form polypeptides or peptides or proteins through a link that's called the phosphodiester bond. That is a peptide bond between the subunits. In the nucleotide case, we have like nucleic acids. These polymer strands form what we know as DNA and RNA in general, and the bond here is called I mistaked. The bond here is phosphodiester bond, obviously. The bond here is a peptide bond. It doesn't have any phosphate in this peptide bond. Sorry for this confusion. And In the case of the monosaccharides, they can form first they can form disaccharides, like we saw in the example last class, and also they can form polysaccharides, like long molecules of glycogen and starch, and so on and so forth.
I actually have a question.
Yeah.
The three bonds that are mentioned here,
Uh-huh.
are they type of the bonds we learned in previous class, or
The-
are they different kind of?
They're all covalent bonds.
They're all covalent.
Yeah.
So why do they have like this?
Because they're special, so they have their own name.
What's special?
Because they are significant. Like a lot of times, if you have something that is repeating itself a lot and you refer to it, then you give it a name, right? Like so, in general, like these bonds have specific properties. For example, the peptide bond has some kind of special form of rigidity to it. The phosphodiester bond is a specific bond that you learn when you learn about DNA synthesis, and has a specific type of enzymes that break these bonds and break these bonds and make these bonds. So you have a whole category of molecules that are related to each one of these molecules. So that's why they have their specific name, but in general, all of them are covalent bonds. Okay. So another question? So the last group that we didn't talk about so much will be the lipids. In this case, this is a phospholipid, so it's composed of a hydrophobic fatty acid, which we'll talk about in a second, what exactly is a fatty acid, and a hydrophilic head. And again, I just told you in short, we're going to get into that in more detail, that because of van der Waals interactions and the hydrophobic effect that we learned about last class, then these effects or these forces stabilize this structure. And this structure is the phospholipid bilayer that is what the membranes of our cell is composed from. Okay, so this is what you have this is actually the boundary of the cell, or the wall of the cell. Wall, I don't like that, but in general, this is like the membrane of the cell. It's composed from a phospholipid bilayer. So I'm just going to skip that because we talked about that. Okay, so fatty acids. So here I have an example of what is a fatty acid. In general, it's just a chain of carbons. Okay, they are attached to one another. Normally, one of the properties of the fatty acid is that it's hydrophobic. Okay, like I showed you in the example. Here I have like olive oil inside this tube, and you can see that it forms these droplets and they do not interact with the water. And this is actually what defines them. This is because they are hydrophobic, so they do not form, for example, hydrogen bonds between these types of molecules and water. And the annotation of these, because they're mostly composed of these long carbon chains. So, for example, this molecule over here, which is palmitic oil, which you can find in my bag. Just a minute. For example, this coconut oil that I have here, you can see it's solid, and we'll talk about that in a second. It's solid it's semi-solid at room temperature, and you can see that this is referred to as C16. So C16 just means that you have, in general, like 16 carbons, and are attached in a row. And the fact that you have 0 here means that all this molecule is what we would call hydrated, or in a sense that it has like hydrogens that are not hydrated in water, like hydrogenized. So in the sense that every one of these each one of these carbons has all is attached to either 2 or 3 hydrogens. Like if it's in the end, we talked about last class that a carbon can form 4 bonds. So either the carbon in the end is attached to 3 hydrogens, and then to the carbon chain, or in the middle it's attached to 2 hydrogens and to 2 other carbons in the chain. And this is what we call in general a saturated fatty acid. Okay, so for example, in this type of oil, we have 93% saturated fatty acid. And an unsaturated fatty acid, for example, glycolate or lake oil, which you can see the example here, is just that, and also the annotation would be the amount of carbons that are not saturated, or the number of double bonds that you're going to have in the molecule is annotated by this number over here. So you have in general 18 carbons and 1 double bond in this specific molecule. And the double bond arises from the fact that now these two carbons, instead of being attached to I'll break this. Instead of having a hydrogen attached to them, they are unsaturated in hydrogens, and they form a double bond between them. And this specific double bond is referred to, and we'll talk about it in a second, is referred to as a cis double bond. And we'll talk in a second about what are the meanings of this specific bond. Okay, so you can imagine from the beginning that what is the difference between a molecule that has this structure and a molecule that has this structure. One of the things that you can see here is that in this representation of this molecule, it introduces this kink inside the molecule, and this kink has, for example, restricts the ability of this molecule to, like, it restricts the degrees of freedom inside the molecule itself. And also what it does is that it restricts the types of interaction that these molecules can have with each other. So for example, you can imagine that if you have a lot of linear molecules, like of these linear molecules, then they can interact with one another in a similar way. No, we didn't see that. But just imagine that you had a lot of these strings or threads that are moving around the liquid with one another. Then because of the interaction that we talked before, for example, van der Waals interactions, a lot of times these molecules can form like a more stable interaction between one another. And this is actually the reason why this is a solid in room temperature. And if you have olive oil that has mostly these kinds of oils inside, will be liquid at room temperature, because the interaction between a lot of these molecules together is not very stable, because the degrees of freedom or the possibilities of interaction between a molecule to another molecule is limited in this case and not in this case. Yeah.
If you heat up like one of these types, can you transfer between the two?
No, in general, it depends how much you heat. Okay, like you can also break it, but in general, like normal heating will not generally change the structure. Like for example, like in a frying pan or stuff like that, you normally not change the basic structure of this.
It feels like one of these should be more stable because like the same atom.
Actually, it's a good question. I don't know like the stability, but like the thermal stability of them, like in response to I can tell you that it resides from, there's a lot of different factors that determine that. Also the length of the molecule a lot of time contributes to the stability, and a lot of the interaction with stuff in the environment. But yeah, I don't know which one of them is more like stable in general, but you can say that these are pretty stable molecules, like they can withstand heat and vice versa. Yeah.
Why does low temperature help the molecules?
Because temperature in general is movement of molecules. Okay, this is like the most strict definition of temperature. Like if you have like more movement, then you have more movement of molecules in the air, then temperature is higher, like or vice versa. So again, imagine this soup of a lot of these molecules together, and the general and everything is like moving around all the time. Okay, so everything is like jittering, and when you have higher temperatures, then the jitter is stronger, and then it takes like a liquid form. This oil, for example, And in lower temperature, you can know that when you put like olive oil in the fridge, probably a lot of you have seen that it tends to solidify. And this is because the lower, like the less movement in general that you have of molecules, then also stable interactions are formed between these molecules, and you get a solid relative to a liquid. Yeah.
And also but unsaturated fatty acids are more difficult to metabolize by the system because of the double bonds.
So actually, the saturated don't have double bonds.
No,
okay.
The unsaturated.
The unsaturated. So I'm not sure, but we have in our body a lot of unsaturated fatty acids. We'll talk in a second. I don't want to, although I really like this material, like in general, a lot of things that are related to cooking. So you'll see a lot of examples to that. But again, this is not. I don't want to talk too much about metabolism and digestion and stuff like that. But we will talk about it in a second when we refer to that. So again, we have either a fatty acid can be either completely saturated or what we will call like unsaturated. And again, it can be a different kinds of unsaturation. Like once it's unsaturated, it can be like one double bond, two double bond, or four double bonds in the molecule. But in general, we refer to this molecule as an unsaturated fatty acid. And because this double bond introduces this spatial rigidity that I showed you, like I showed here, then this spatial rigidity can exist in two ways. Okay, so you can either have what we would like to call a cis bond, a cis double bond, which you have the hydrogens in the same side, so the cis side, or you can have a trans carbon double bond. Okay, so in this case, you have the hydrogens on the opposite sides of this double bond, and this is referred to as trans. So a lot of you probably heard the concept of trans fat, right? So this is exactly what this means. And again, this is just summarizes what I said before, that there's two stereoisometric configurations. Like I told you, like stereoisomers is when we say that a molecule has exactly the same composition, like molecular composition, but just the order of the bonds is different. So because this introduces rigidity, then you can have this conformation of the cis and trans. And a trans double bond is very rare in nature, and not very rare in your margarine, okay? Because actually, it's very interesting, like I talked to you before, and I'll also give an example in the next chapter, about that sometimes we think like, what's the big difference, right? It's just like, I showed you something like this, and now it's something like this. So, but actually, this form is very rare in nature. Like, apparently, there is something like the enzymes that are producing these reactions are probably like non-existent, or prefer to just form one type of molecule and not the other type. But so where does this come from in a margarine, for example? So in margarine, as you know, you take you take a plant-based fat, for example, like olive oil in a good case, and then you hydrogenate that molecule. So you add hydrogens to that molecule in order to make it saturated. And because it is saturated, then again you can achieve this rigidity that a lot of time people are looking for, and that's a big difference between margarine and just the liquid form of the vegetable oil that it's formed from, that it's just solid. And that's why you can form all these pastries and things that are not tasty from that. So, but the side product of that of this process is you can also form this trans double bond. So, although it doesn't look like that, this molecule is actually very stable with one another. So, if you have a mixture of these molecules together, so not only that they are now linear in form, which means that they can interact with each other so I can illustrate it with this. For example, if you have all these are linear molecules, so they have a lot of possibilities to interact with one another, okay? And because they can have close proximity between the molecules themselves, then it allows van der Waals interactions like we talked before, because van der Waals is very much dependent of the distance, okay? So you can imagine that if I would have a kink in this molecule, then it would limit the possibilities of van der Waals interactions, and thus it will be more liquid. So here, not only that you have this linear form because of the cis bond, you also lose the rigidity that you had before in this axis, okay? Because you have a double bond now, and this double bond cannot twist and turn, and it's much more rigid. So this tends to solidify much more, but the problem is that this is completely artificial and apparently very bad for your health, okay? So, there's a lot of people that are talking now about a lot of time people are referring to saturated fat as they're referring to trans fat, but this is actually very bad press for saturated fat, because saturated fat is completely natural and this is completely artificial. So you don't have this in nature, and I guess that a lot of you will not like to participate in an experiment and to eat something that was never, is not common in nature. So this is just a chart of different fats. You can look at it later. I'm not going to go over that, but the amount of saturated fat that you have in saturated and types of fat that you have in several oils, olive oil. That, for example, you have most of it is oleic oil, and you have very low amounts of the other fats. And this is just a fun fact in the consumption to show you how much publicity has to do with these types of things, and how now today we know much better. And this is why you see now that just in the past years, in 2010, finally butter is starting to pass margarine in the sales in the U.S. So in the 60s and the 40s there was, or 30s, there was massive publicity against butter and for margarine, which was considered much more healthy because it's a vegetable, right? But this just shows you that you need to know chemistry, okay? Yeah.
So comparing trans and trans is more stable or more?
Yeah, trans is a much more. It's stable in the sense of not inside the molecule, like I was explaining before. It's stable in the sense that it tends to interact more and aggregate more, also in your veins. Okay, so apparently in your blood vessels it tends also to aggregate more.
It's just nickel, but I can't believe it's not butter. It said no hydrogenated.
Yeah, because this is a new version of this. So now they make it. I don't know which process they're doing, probably something else, no hydrogenation in the process, because a lot of times people tend to fixate on words and catchphrases. Okay, so people have heard that hydrogenated oils are bad, so they add no hydrogenated oils. So now they do it with some kind of other chemical reaction or add something that causes it to solidify. I don't know what.
And no trans.
And they say also no trans fat. Yeah. So now they make it so this product was very different in the 40s and 50s. It was pure margarine with almost a lot of it was trans fat, and today they just make it from something else. I don't know. They found another. I don't know what is the chemical process that they now make margarine, but a lot of the margarines that they make today doesn't have trans fat or hydrogenated oil, but I don't know how they make it.
What's the difference from butter?
Butter is saturated, basically a saturated fat. Most of it is just.
Yeah, but if they do saturate the unsaturated fat, then they become saturated.
Some of them become saturated. So if you have unsaturated fat and you saturate it, so you can either you get both of these products.
Yeah, but I mean, if they say that in margarine, this specific margarine, they take unsaturated fat
So,
configuration, then what they do get is butter.
No, because it's a different oil.
well, I think that in this case they don't saturate it. They do something else. Probably they add some kind of other chemical that stabilizes this structure, and not through this process anymore. Okay, but we need. Okay, so in general, this was a very long introduction about lipids, but in general, lipids are not. You will not see them in this form, like in the body. In general, they are formed in the form of phospholipids. And most commonly in which that the phospholipids are made from a fatty acid chain, like I said before, and this, for example, is a double bond, okay? It doesn't tell you if it's, like here it looks like a trans, but, but it's not necessarily. So, you have, in this type of molecule, in this type of phospholipid, you have a fatty acid chain part, which is attached through a molecule, through a glycerol unit, and a phosphate to the hydrophilic head. Okay, so this molecule is what we would term as amphipathic, which means that it has also hydrophobic and hydrophilic parts. And this part interacts with water, and this part does not interact with water, like I showed you before. So these amphipathic phospholipids can aggregate or can accumulate in the cell in different forms. The one that we're going to focus most about is obviously the phospholipid bilayer, in which there are double layer with the hydrophilic area facing towards the water, that is inside the cell and outside the cell, it doesn't matter, and the hydrophobic area that is in the center. And again, this is stabilized through through the hydrophobic effect and van der Waals interaction between the chains themselves. And liposomes, which a lot of times are used for secretion of certain molecules, and also for proteins that you want that the cell is shipping to the membrane. We'll talk about that later. And also micelles, which are just monolayers that aggregate together of fat molecules.
Micelle looks like more steady.
It looks, but it has to have very few amounts of it has to have relatively less amount of molecules in order to form a micelle. You cannot form like a micelle. If I would add much more molecules here, then the micelle will not be able to form. You could have several micelles, but then you have the problem of the hydrophobic effect that I talked to you before, that if you have like a droplet and two droplets are next to one another, then the thermodynamic stability would push them together. So a lot of time. Yeah, you're actually right. That was wrong what I said now. But no, it's good that you asked that. But in general, I think that the reason that you don't have these micelles is that in the cell you have a lot of different types of proteins, and you have a lot of different types of scaffolds that are holding all these structures together. So it's not that this type of structure is like free in space. Okay, it's mostly stabilized, but a lot of anchor proteins are keeping it together. But you're right, it was a good catch. Okay, so in this we finished the part about the chemical. It's the opposite direction. The chemical building blocks of the cell, and I will talk a little bit about biochemical energies. So, in biology, the relevant forms of energy is thermal energy, mechanical energy, electric energy, and we'll mostly focus on chemical potential energy and concentration gradient energy. Specifically, when we're going to talk about like transport of ions across the membrane, we'll talk a lot about concentration gradients. So cells can transform one type of energy into the other type of energy. For example, in photosynthesis, which is the nicest example, there is a transformation of the sun's energy into chemical energy that is stored in the form of sugar. Okay, that we eat, or we eat the animals that eat these sugars and know how to make energy for the function of the cell themselves. So, question? now we're going to go over like a basic course in thermodynamics in one slide. And according to Gibbs' law, all systems change in such a way that free energy is minimized. And I mean free energy of the system, not free energy like in general. The amount of energy in the world is constant. Okay, and it just it changes in a way that free energy is minimized in a given system. And specifically, what do I mean? Like, if you have this equation, the change in energy is derived from the change in energy between the product and the reactants, and is also given by the equation in which the change in energy is determined between the change in this is called enthalpy, or in our case, the bond energy, minus the temperature in degrees, which is constant in our case normally, and the entropy. So it's okay if you don't understand that. We're going to give a, like, yeah.
What do you mean by free energy?
So I'll give an example, and then it will be like more. I know that this is a very problematic concept in general, and there's no way to explain it in like one slide. But I'll try to explain through the examples. And the thing that I want you to remember from this slide in general is that there is energy in bonds or in molecules. That this energy can be released with breaking of these bonds, and can be harnessed or transferred through the breaking of these bonds from one molecule to another in order to form functions, cellular functions in the cell. So in general, an exothermic reaction is one where the products contain less bond energy than the reactants. Okay? So the products contain less energy of the reactants. The delta G here will be negative, and it will be called like an exothermic reaction that releases energy into the environment. And vice versa for endothermic reaction, meaning that the products contain more energy than the reactants, and it makes sense that you need to invest energy into making these products relative to the other way around. For example, one of the most common exothermic reactions is the burning of wood, which is generally just breaking of most of what wood is composed of, like I told you before, is like all types of polysaccharides or sugar in general. So the breaking of sugar together with water and the investment of initial energy causes a release of energy in the form of heat, in this case, and the formation of the products, which are mainly CO2 and water, and also byproducts of the burning, which are ashes, generally, like the compound of a lot of different types of molecules, obviously because a lot of times this process is not strictly like pure sugar. So in this example, the bond energy or the energy of these products is lower, or it contains less bond energy than the reactants, and in this case you have an exothermic reaction. Okay? Questions? So, yeah.
So why do you have to invest energy?
You have to invest a little bit of energy in order to initiate like to get the reaction started. So in this case, you have to like apply a match or light a lighter to give a little bit energy for the reaction to occur, and then because the reaction is exothermic, then it's feeding itself. Okay, once you invest the initial amount of energy in order to activate the reaction, then it happens spontaneously and releases energy, and the energy that is released is used for the continuity of the reaction in other molecules of the wood. So you have to just ignite like a small piece of the wood, and then this catalyzes the continuation of the reaction. Okay?
So the energy is the strength of the bond?
The energy is not strictly like the strength of the bond. I don't want to get too much into that. We will learn about specifically about one type of bond that that the release of that bond or the breaking of that bond contributes energy to the system. And we'll talk specifically about that. I don't want to get too much into that because obviously this is a very complex notion. Just to let you know that you can also derive for a specific type of reaction from the K equilibrium that we learned about before, and these are just constants. In general, then you can derive directly the amount of free energy that you will get or invest, or you have to invest in order to perform this reaction. Okay, so remember that the K equilibrium is made from the so here I explain it in the example of ATP. What time is it? Why don't I have just a minute? What time is it? Okay. So I have time. So ATP is, you can say, like, I don't like the term most important, but it's one of the central molecules that are used by the cell in order to capture and transiently store energy and and convert energy or use this energy in order to perform different types of chemical reactions, okay, in the cell. So this is how this molecule looks like in general. So it's made from adenosine, which is a base, which is a nucleotide that is composed of an adenosine base and the sugar, and 3 phosphates that are attached through a phosphohydride bond over here. So these bonds contain energy, and once they are broken, or this phosphate is transferred, for example, to another molecule, then it gives it energy that it later can use in order to perform a reaction. For example, if we have substance B together with ATP, that has ATP with 3 phosphates attached to it, then this phosphate can move and attach to this B and give this energy in order to produce from B and C the reactions, the product, which is D, and the free phosphate that is released in this case. Okay, so in this case, in this example, we have ATP that is giving this reaction the energy that it needs through the formation of this B attached to this phosphate. Okay, so the transfer of this phosphate that is occurring here gives the energy for this reaction to occur here, and because of this release of this phosphate bond, that's the formation from B and C substance D. Yeah. What kind of energy is released? Like general? Yeah, it's mechanical energy. Like that is just forming covalent bonds, for example, between 2 molecules in general. Okay, so let's say you have 2 types of molecules and you want to bind them together through covalent bonds, then the energy that is released here will be invested in the bond that is formed between these 2 molecules. Okay? Because you need, for example, if you want to construct something, you want to make something, you need to invest energy, right? To hold, like, if I want to, like, form this reaction, then the breaking of the phosphate here would give the energy that is needed in order to connect these 2 molecules together. And this will be like a higher energy product than this form. Okay, so remember that we, for example, in the form of the wood that we talked before, this form has more energy, more free energy than the subunits, for example. So if I want to, so this is not a very good example because it's not wood. It's not true for any molecule, but in general, the energy that is released here is used as a mechanical energy in order to. Yeah. Like you can. It stabilizes. We'll actually learn specifically about that when we talk about enzymes. But yeah, in general, the energy here, like you can say, like sets the conditions that are favorable for this bond to occur. Okay.
You can also break bonds, right?
Yeah, you can also break bonds with. Yeah.
The energy transfer is from ATP to the B plus CI.
there's also energy transfer from here to here, but a lot of times like these, in this, the sum of the free energies that is being released is not so high. And mostly what I'm talking about here, like the major amount of work or the major amount of energy that is being released or being invested is in this transition. But this is just like a general example. Okay.
I know, but I'm not understand like how much spontaneous ATP
would break. So it does break, like theoretically it should break really, it's a really unstable molecule in general. But the thing is that the cell does not keep like a store, like a general store of ATP. A lot of time it makes it near the locations where it has to be useful. Okay, so you have like the mitochondria that is making ATP. In general, we're going to talk about it in the next classes. But it has to produce ATP all the time. Okay, in order for it to make to be available. For example, you don't have like you have mitochondria everywhere in the cell. Okay, you have in synapses, you have in like axons, you have everywhere in the cell. You have mitochondria because of the instability of ATP in general. So this is just in numbers. I think I'm going to skip that. You can look at it later. To how so this is general, and this is a very general amount because it really depends on what ATP is reacting with in order to know how much free energy is being released through the hydrolysis of ATP. So ATP plus H2O can make ADP or diphosphate. So this is adenosine triphosphate and this is adenosine diphosphate and a free phosphate. So in this reaction in general, you have between 35 and 40 kilojoules per mole of free energy that is being released through this hydrolysis. Okay? So you can go over it later and also see a more detailed explanation here. So, again, I would like to say because this topic is very confusing and it's very hard also to grasp this concept, the most important thing is to understand that ATP is important for storage of energy and is readily able to transfer energy and catalyze certain energy that is needed in order to perform a lot of cellular functions. And that's it, basically. So, we finished this chapter. For next week, you need to read the relevant chapter in Lodish, which is chapter 2, and go over the questions for the chapter that we'll go over for next week. So how much? I have a few minutes. That's fine. What?
Yeah, maybe it's like not exactly.
Here it says 44, so I have 1 minute. Okay, so I'll just give you the example that I gave like a lot of time before this chapter about another stereoisomer, which is the thalidomide, just to show the importance of the specific order of a chemical bond inside a molecule. Have you heard about this example of thalidomide? That during the 60s or 50s, generally this was subscribed, this material, the R-thalidomide, which is the only difference between them is as you see like the orientation of this bond. Okay, so in one of them is like in this orientation and the other one is this orientation. Okay, like this ring over here. So the R-thalidomide is actually effective treatment for morning sicknesses and was sold. And this compound, like the thalidomide drug, which normally composed of a combination of these two molecules. But the problem is that this molecule is a very effective treatment for morning sickness, but this molecule causes severe birth defects. So there was a big, it caused like the birth of a lot of children with severe deformalities because of the treatment of this specific drug. So it just shows you that we people need to be very careful about the chemical reactions that we do, like in the case of thalidomide, and less severe case in the case of margarine. So a lot of time when you see like a molecule, the compound And the actual order and the shape of the molecule is very important, a lot of time more than the actual molecules that compose it or make it. Okay? So we'll meet in 15 minutes or 14 minutes. יש את הקבוצות משנה שעברה, השנה לא ראיתי לאן הם העלו אותם. במודל, ראית את הקבוצות משנה שעברה?
יש באחד שקופיות הראשונות מצגת מקודמת שלך, כמה תקנים שהיו.
כן, לא שמתי את זה במודל?
שמתי את זה במצגת.
כן, חשבתי ששמתי את זה בנפרד. תראה, אני מלמד את הקורס הזה שנה שנייה, לפני זה היה מרצה אחר, זה קצת לא כל כך רלוונטי, כאילו זה אותו חומר.
לא שומעים, אני לא האמת לא ניסיתי את כל ה...
ב-2015 לא שומעים?
כאילו לא הצלחתי להעביר בין השנים, אפשר להיכנס.
כן, כן, את יכולה...
יש דרך לקבל את הקבוצות עכשיו, שיש דברים ש...
כן, אני אראה כאילו איך אנחנו מארגנים את זה. את יודעת מה, אני בתחילת השיעור אני אראה את זה במודל. כן. בגלל שבעצם הראינו שבמצב הזה, אז הריכוז, בגלל שה-pH שווה ל-pK בעצם ישירות מהישרה הזאת, אז לא, הריכוז בין שני הספרים האלו הוא שווה. ואז, כן, בין יותר החומדה בבסיס המצומד שלה. זאת אומרת שיש לך את הדבר הזה בבסיס עכשיו, ברגע שהוא משחרר את המימן שלו הוא כבר... זאת אומרת שיותר מדי על המונחים האלה, אבל בגדול, מה שאת יכולה לראות מפה זה שלמשל אם יש לי, זה הדוגמה יותר מסוימת. בואי נגיד שאנחנו נמצאים ב-pH 2, ואנחנו יש לנו את החומר הזה. אז אנחנו יודעים, יש לנו את החומר הזה בתוך התמיסה שלנו. בהכרח אנחנו יודעים שאנחנו נמצאים במצב ש-H3PO4 ו-H2 ו-H2PO4 הם נמצאים ביחס של 50-50 אחד לשני. שיש 50% מהחומר נמצא במצב הזה, 50% מהחומר נמצא עכשיו, מה שזה, זה פשוט נובע למשוואה מבחינת ברגע שהפיק זה שוב משהו שאנחנו יכולים לחשב אותו ישירות מהקבוע של משקע. את יכולה לדעת, את יכולה לכתוב באינטרנט רשימה של מה הפיק כמה זה יפה. אבל ברגע שהפיק שווה לפיק והכמות של החומר של החומצה בסיס המצומד שלהם שווים, אז זה בעצם אומר שאם אני אוסיף עכשיו מימנים, למשל תוסיף חומצה ויבספו את מימנים, אז יהיה לך הרבה, כאילו 50% זה כאילו נחשב כאילו פרקציה מרכזית שיכולה להגיב איתו ולהמיר אותו חזרה למצב של חומצה. ואז כאילו לכלוא את המימנים האלה ולדחוף את הריאקציה לכיוון הזה. כי בסוף שם מימנים, אבל בעצם מה שקורה זה שבגלל שיש פה עכשיו הריכוז של המימנים הלך וגדל, בגלל כמו שזה משקל, ברגע שאני מגדיל את הריכוז של משהו אחד, זה דוחף יותר לכיוון של בגלל שבמקרה השני, אם נגיד יקרה מצב שתוסיפי בסיס ואת תשווים מימנים, אז החומצה תשחרר עכשיו את המימנים ותהפך להיות בצורה של המצומד שלה. אבל כל הזמן ה-pH יישאר יחסית לזה. בעצם במצב 50-50, תחשבים מצב קיצון, שיש 90% ו-10%, אז מספיק שאני מוסיף טיפה מימנים, אני מכלה את ה-10% האלה שיש לי אז זהו, זה כבר לא אפקטיבי וכל מימן שיתוסף פשוט יעלה לי ישירות את ה-pH. כי המימן זה pH זה פשוט הריכוז של המימנים שיש. חומצה שהיא מאוד מאוד חומצית, אז יהיה לי מאוד קשה להפוך אותה להיות בסיסית. אבל אם אני אוסיף... נכון, אז אני רוצה בסופו של דבר שיהיה לי pH שישמר לי סביב ערך מסוים. אז נגיד אם יש לי חומצה, אם יש לי נגיד ה-pH שנמצא באזור 2, זה pH מאוד חומצי, אם אני אוסיף חומצה, זה יהיה עוד יותר חומצי. וזה יהיה עוד יותר חומצי מהר, בגלל שלא יהיה לי שום דבר שיסתור את חומצה מהצד השני.
ה-pH ירד ושתתן לחומצה?
כן, ה-pH כאילו ירד והוא לא יתאזן ל-2. אבל במצב שיש לי שגם החומצה וגם החומצה המתאימה שלהם נמצאים ב-50% מהריכוז שלהם, במצב הזה צריך לחשוב את זה כאילו שכל אחד מהם פשוט יכול מאוד בקלות להגיב עם החומר שהוספתי עם החומצה או בסיס ולהחזיר את ה-pH למצב שלהם.
זה לא אומר שהוא יכול להיקצר בקלות, זה אומר שהוא יכול פשוט לשמור.
כן. זה גם אומר שיכול להתקשר, שמגיב מהר עם המימן או זה ש... אבל... כן. כן. את יכולה למשל בפורום, אם זה לא... זה. כאילו מה ש... Yeah.
My name is Hi. I'm sorry, so actually I haven't been at this class last week.
Okay.
Because of my actually my flight ticket to Israel get some homework. Okay. So actually my bachelor degree is informatics. I already learned some basics about biology and biochemistry. So I'm wondering if I can do this course.
Well, the thing is that I normally say it depends how much general physiology, because it's good that you, in a sense, miss the first two classes, and also this class. We're going to talk strictly about things that anyone that did some type of degree in biology is supposed to know, even if it's bioinformatics. But a lot of people did not do the more physiological part. That is talking about channels and ion channels and concentration gradients across the membrane and action potential and how more molecular neurobiology, for example, like RNA, neuronal RNA and RNA regulation and nervous system. Stuff like that. Normally, people that did a general course in general degree in biology, in basic biology, don't cover that material.
Okay. So you think I had better to...
It depends if you did physiology. If you took physiology courses and neurobiology courses, then maybe it's not relevant. But if you didn't take these courses, then I would recommend that you would miss, you can miss this class and the next class, and then just come for in 2 weeks from now, and then we're going to talk about things that are more relevant to neurobiology, not general biology.
Okay. I understand what you mean. So, and about the food that you recommend? About the food you recommend?
Yeah.
Yeah, so can I find...
You can find it here.
Can I find it in Moodle?
You can find it here.
So, do you have a PDF version?
The PDF version is not you can find generally a PDF version, but I'm not allowed to distribute it because it's copyright and stuff like that. But it's also in the library here. They have a few copies of the book, and also in the library, the main library. Life Sciences also has copies of that book that you can take. But you can also find between us. You can find the PDF version online. Okay, so have a seat, please. So, in the next chapter, we're going to learn about protein structure and function. Again, like I told people that have a basis in biology in general, this is not required for you to take to participate in these lessons, because again, this is very basic knowledge for anyone who is taking anything in biology. This is based on Lodi chapter 3, so again, you can look at it in the actual book and see if you're missing information, then decide if you want to go to the following. So it will be, I will try to finish it in this hour, then next week it will be 2 full hours, and then the week after it will be 1 more hour. So like in the middle of the class in next 2 weeks, we will probably finish this chapter, which is a large chapter. So in this chapter, we're going to go over the hierarchical structure of proteins, folding, modification, degradation of proteins, talk about enzymes, molecular motors, also in the sense of neuronal systems, and mechanism of regulation, and a little bit about methods that people are using in order to purify and characterize proteins. So, in general, the molecular structure, or the organization, or the levels of hierarchy, when we refer to proteins, are primary. We refer to it as primary sequence, which is the actual backbone of the protein. So if you have a linear form, this is like a short peptide that I made here from molecules. So this will be the primary structure. The secondary structure is what we will call local folding, and specific structures that are formed in close proximity or close range of protein folding will be the secondary structure, and we'll talk about the special types of formation that you have here. Then tertiary structure, which is long range folding of the actual molecule. So in all these levels, we're actually talking about the same molecule. And same molecule, I mean that is bound together with a covalent bond. Okay, so for me, it's very important to make this distinction because it can be very confusing. And from this level on, we're now talking about multimeric organization. So several subunits, for example, of the same protein, which can be composed of different molecules. Okay, so one protein does not necessarily need to be and a lot of times is not referred to as one molecule. It can be composed of different units of different molecules. So these are basically the main the major hierarchies when people are referring to as protein folding. And there's also supramolecular organization, but this is a very vague definition, because it just is the general interactions between different proteins and different systems. So you can also do supramolecular or whatever. So this level is very vague in a sense, but all these other levels are very well defined. Okay, so in a sense, this is where the protein stops, in the quaternary level, the definition of a single protein. So obviously, the function of protein is derived from their structure, and their structure is a three-dimensional structure, or the way that they fold in space. And this folding is specified and is determined by the sequence of the amino acids. So, like I was asking before, different amino acids tend to fold in different ways, and and although there are different, we'll talk about it a little bit when we talk about energies of folding. But in general, the three-dimensional structure is determined by the sequence of the amino acids. So a different sequence will, most of the time, give you a different structure, okay, of the protein. So the primary structure of a protein is just referred to as the backbone of the protein, and it doesn't attribute any folding in this level. So when you have a sequence, like I showed you before, a sequence of amino acids that are bound together, it is referred to as a peptide or a protein. It depends on the length. And this strain or this backbone has a directionality that is normally referred to as C-terminal. So in this example here, we have 3 amino acids that are connected to one another. The green here is the side chains. So normally the side chains are not small. You will see, like, I'm just simplifying it. But in general, this is the basic backbone of a peptide with 3 amino acids. And here in the purple, which here are in yellow, I marked the peptide bonds that are linking between different amino acids. Okay? So this is one amino acid, this is a second amino acid, and this is a third amino acid. And you can see that one side, in this case now, one side has the C-terminal because of the carbonyl side, and the other side, the amide or amine side, is the N-terminal. Okay? So this has directionality, and the last time is very important because several proteins, especially channels, tend to have one side that is sticking outside the membrane and the other side that is sticking, for example, intracellularly. So this is just a linear arrangement. And in general, here's a good place to talk about sizes of general proteins. So a protein, this polypeptide chain can be as many as 4,000 residues. But this is like the more or less the maximum size of various proteins. And short polypeptide chains are normally referred to as peptides. Okay, so everything is a peptide. Also a protein is a type of peptide. But in general, when you will hear about peptides, they're usually up to 20 amino acids will be referred to as peptides, and if they're longer, they will be referred to as proteins. Okay? And the typical weight of an amino acid is between 75 and 200 daltons. Which dalton is just a unit that we use in order to describe the weight of a lot of molecular elements, and specifically it's between 75 and 200 daltons, typical weight of a single amino acid. Questions? Yeah. So, secondary structures are the spatial arrangement that results from local folding in the polypeptide chain. So in general, you will not find this linear formation inside the cell, okay? Because this linear formation is not very stable. It tends to fold on top of each other, and I can't show you because I only have 3 amino acids, so I can't even show you the most basic secondary structure. But in the cell, mostly stabilizing hydrogen bonds, and here it's important to remember that the hydrogen bonds within the actual proteins are the ones that are stabilizing these structures, tend to form well-defined periodic structures that are named alpha helix, beta sheet, or short U-shaped turns. So the first structure that we're going to talk about is the alpha helix, and here you can see like the backbone of the peptide that is marked here in red, and the R groups, which are just the side chains. So again, I don't want you to imagine that all the side chains are like the same in their balls. Okay, they're complex molecules, a lot of times larger than the actual backbone, but for simplicity, in order to show this formation here. And what you can see is that the hydrogens are in hydrogen bond affinity, or form hydrogen bonds between the oxygens of amino acid and like amino acids inside this backbone. So in general, you have 3.6 residues per one helical turn, and I can't really show it here in an exact way, but you can imagine, for example, from here that you have one of the hydrogens, for example, that is connected to one amine in this case, that can like form a loop and stabilize with oxygen on the other side, and this formation causes a stable helix. Okay? And what is unique about alpha helix is that all the hydrogen bonds, all the hydrogen bonds inside the molecules are attached or accompanied. So you don't have, once you have an alpha helix formation, you will not have free hydrogens that are able to form new hydrogen bonds. And because of that fact, you can imagine that it will tend to be non-reactive, like the actual backbone will not form interactions with water molecules. And thus, the hydrophilic or hydrophobic quality of the helix will be determined entirely by the types of amino acids that you have inside the backbone itself. Okay, so if you have hydrophilic side chains or amino acids, then this whole helix will be hydrophilic, and if you have hydrophobic, so this whole helix will be hydrophobic. Okay? Question?
Yeah. In this case, the residues don't interact, also don't change the structure. It's only the backbone that's making the structure.
In this case, no. In this case, it's like a very local folding. It really resides from the basic structure of the backbone itself, and these interactions are inside the actual backbone itself. It does not involve. Obviously, the side residues have. We'll talk about it in a second. They have not all, for example, the identity of the side chains can have an effect to whether this amino acid will be part of an alpha helix or not. So you have, for example, specific types of amino acids that because of their unique side group cannot twist in the way that is needed in order to form an alpha helix. Okay? And we'll talk about it in a second. Yeah.
I'm not sure I understood what you said. You said that there's hydrogen bonds which affect the reactivity.
So all the hydrogen, we learned that hydrogen bonds are the main connection that water interacts with substances inside the aqueous solution. So in this case, if I have like a linear peptide, then I have a lot of hydrogens here that are able to form hydrogen bonds with water surrounding it. But the unique case of the alpha helix is stabilized by all the hydrogen bonds inside this structure. So what it does, it occupies all the hydrogen bonds inside the backbone in a way that they're not free to interact with water anymore. So let's say we have that all the side chains here will be hydrophobic. Then this alpha helix will be hydrophobic because you don't have any component or any atom that is able to interact with water. And if all of these will be hydrophilic, then, or some of them will be hydrophilic, then it will be hydrophilic because they will interact through the side groups. But it will not reside from the interaction will not happen with the actual backbone of the chain.
So only the residues will affect.
Yeah, that's the most important thing. Like to remember is that in alpha helix formation, the residues are the ones that determine the hydrophobicity or hydrophilicity. Okay? So, because this is molecular biology, I will try to accompany like a lot of these, again, like step aside for a second and say that I think that in order to understand like molecular biology in general, in order to understand all these, you have all these chemical interaction and everything, you have to have a very good imagination. Okay? You need to imagine yourself like stepping into this environment and seeing how these molecules are actually behaving. And because of that, I will show you a lot of videos. I try to, also because it can be hard sometimes. So I will show you a lot of videos that will help you develop your imagination a little bit. I'm not hearing anything. I think there's sound, but for some reason. Never mind. So this is just an illustration of how a general alpha helix looks. From the linear structure, you see it like twisting. And for example, and this is just illustrating the actual backbone. So a lot of times in articles and in certain figures, the only thing that you're going to see is this ribbon formation. Okay? But what actually is there is the thing, it's what you saw before. Okay? What is actually there is the actual molecule. So for example, this thing is a side chain of the amino acid, of this amino acid. And here's another side chain of this amino acid. Okay, so this is the thing that I didn't show you before. And these are hydrogen bonds that are formed between, again, between oxygen and the hydrogen that is not shown here, that is attached to this amine group in this example. Okay. So, next structure is the beta sheet. And in the beta sheet, you can have, it's composed mostly of short 5 to 8 residues that are folding on each other and being stabilized again by hydrogen bonds. Okay? So remember that I told you about the hydrogen bonds that are linear and non-linear. I don't know if you remember that, but in these cases, a lot of time in beta sheets, you have non-linear interaction between, or non-linear hydrogen bonds. And what is important to remember is that a lot of times the hydrogen, this type of folding can also be like, it's referred to as a beta sheet, but it can actually reside from residues that are very far from one another in the actual polypeptide chain. So for example, here, this strand can go like here, and then return like this way, and then go here, and then make loops here, and then return to here. But in general, the structure here, the beta sheet structure, is stabilized by hydrogen bonds. But here you can see that some of the hydrogen bonds, or some of the hydrogens, are available to interact with water. So a lot of times, excluding the case of a very special type of beta sheet barrel that we're going to talk about a little bit, then excluding that example, this will tend to be more hydrophilic than hydrophobic because of the interaction of the hydrogens in this structure.
Because it's already using the hydrogens to...
So it's not using all of them. It's using part of them in order to stabilize the structure, but relative to the alpha helix in which all the hydrogen bonds are occupied, here not all of them are occupied. Okay.
So you can interact more with water.
Yeah. So it has more possibilities to interact with water.
What do you mean by the hydrogen bond?
So for example, here you have a hydrogen that is not coupled to anything.
No, I mean the hydrogen bonds are not linear.
Here. So for example, you can have like shifts. It doesn't have to be like straight between these two lines. You can have like a shift in the linearity, and that's an increase in the length of the hydrogen bond. So what I thought, yeah, because it's like diagonal. But again, it's not a very important point. Question? Yeah.
Can you just elaborate on what causes the peptides to form those structures? Why would a polypeptide form one or another?
So it really depends on the identity of the amino acids, because certain amino acids will tend under certain conformations, and also the identity of the amino acids, for example, in the terns, will determine if it's energetically favorable to form these structures. Okay, so in a second I will talk about the actual angles that you have inside each amino acid, and because of these angles tend to form the structures which are mostly beta sheets and alpha helixes. But actually the identity and the combination in the sequence of these amino acids will determine, because this is like more a local folding, except from this example, but mostly the identity of these types of amino acid will determine whether it will form a beta sheet or a alpha helix, for example. Okay? So this connects me directly to the concept of the phi and psi angles that are inside a peptide backbone. And these two angles, for this I need my, my model. So these two angles generally are defined as the plane that is formed between the carbon. So this is my alpha carbon. The alpha carbon is the one that is attached to my residue. Okay? So the phi is defined by the plane that is formed between this, this, I'm looking at it the other way around. So this carbon, this amine, alpha carbon, and this carbon. So this, right? Yeah, I think it's right. No. This is the phi. Ah, yeah, it's okay. So this is the phi angle that I have here. Okay? What? There's a, because I may be mistaken. It's very hard. So this is the phi angle. Actually, I'll hold it like this. It's hard to see, but again, it's very hard to imagine really without the models. But in general, if you imagine like a plane that is going something like this, then this twist is along the phi angle, and this twist over here is along the psi angle. Okay? So there are two major angles that determine inside each amino acid that will determine the actual orientation in space of this molecule, okay, of the amino acid. And because of this orientation, then you can get typical angles or combinations of phi and psi that will produce structures of alpha helixes or beta sheets, okay? And the third angle, for example, omega, is normally has less maneuverability because of a special, and this is just a side note, because of a special resonance relationship that occurs between these three, between the oxygen, the carbon, and the amine group over here. So this introduces actually a rigidity to the omega bond. So this is why we normally do not talk about this type of bond, and we just refer to these two angles that are around the center or the alpha carbon that we have in the middle. Okay.
What is the other group? What is the third group that's not the
The third group is the U-turn, which we'll talk about in a second. The thing is that U-turns are much less well-defined, than the alpha and beta. They can be all sorts of orientations, and also they're very short, just a turn of a few amino acids. But we'll talk about it in a second. But so what's important about this plot is that this is experimental. Okay, so here you have like thousands of proteins that were measured, and each point is the actual confirmation of the angle. This is taken from crystallography. We'll talk about it when we go over a crystallography article. But from a lot of crystallography experiments, they actually measured what is the angle. So each dot here is an amino acid, and this describes in which orientation or what is the combination of phi and psi that this amino acid is in. Okay, so each amino acid, like I told you, can undergo several orientation or several combinations of phi and psi, and this is determined a lot from the environment and the general folding also of the protein, but it can also be determined directly from the type of side group that this amino acid has. So, for example, you can imagine that if you would have a specific amino acid that will have like a huge side group, like this, then you can imagine, so this is the side group, then you can imagine that this will impose difficulties and will restrict the possible angles that this amino acid can be in because of steric interference. So again, in the imaginary part, don't think about it as this. It actually, everything here is surrounded by thousands of forces of attraction and repulsion between electron clouds of these atoms with one another. So the actual size of this is like, you can imagine that it's like all these huge balls that are sticking around, because this is not like neutral in space. Like each one of these molecules is actually sensing all the other molecules. It has like electrostatic interactions with everything. So it's not that it's possible for this molecule to like get inside the other molecule and twist and form a knot or something like that, because the steric interference is just too large. Okay?
What was it? Proteins that don't fall into either, so what are they?
So in most proteins, so again, this is a resolution of amino acids. We're not talking about the proteins, but in general, you will find alpha helices and beta sheets almost in any protein. Okay, because as you can see, most of the amino acids have the possibility to form either these structures. Okay.
So the rest, they don't have the form.
So a lot of times here it's just an amino acid that, because of the conformation of the restriction that the actual backbone imposed on this amino acid, it is not, the fine psi is not part of an alpha helix or a beta strand.
But that can't be lots of amino acids because there are like 22.
No, but there's a lot of proteins.
Yeah,
So
these are proteins.
yeah, so these are, it's a resolution of amino acids, so each spot is an amino acid. But let's say I would want to generate this for, I don't know, I would take a protein that has like 200 amino acids. And I will measure for each amino acid in that protein what is the fine psi of each amino acid, and I will put it on a plot.
So an amino
So-
acid can have a different,
Yeah.
the same amino acid
Yeah.
can have different phi and psi.
Yeah, that's a very important point. Okay, a certain amino acid does not have to be necessarily in an alpha helix or a beta. It can also be like in the center. And it can be like part of a U-turn, but not all of them. And we'll talk about it right now. So, yeah.
And it's like the only typical plot.
The Ramachandran plots are used for a lot of different things to describe like relationships. It doesn't have to be like proteins.
Only this one.
No, but in general in proteins, this is the major, like most proteins, most amino acids would either be in alpha helical formation. In a protein will be either in alpha helical, in beta sheet, or in U-turn formation.
Do you have an idea why you use like mostly negative phi instead of?
I think it's I don't know how they defined it. Like
Can you just turn it around and make it
Maybe it's not only convention.
yeah, I think it's I don't know. I think it's physicists. I don't get into their mind. Anyway, so here we have all 20 amino acids and a typical Ramachandran and like a heat Ramachandran plot for each one of them. And this is just showing you like the typical or the more energetically favorable orientation or combination of psi and psi that each amino acid can have. So we talked a little bit about special amino acids. Inside of them one of them was proline. So in proline, for example, we have this special formation. It's not actual proline. I just show you for illustration, in which the side group is actually connected to the backbone through a carbon chain. So you can imagine that this will impose restrictions on the angles of the phi and psi that this molecule can have. And the other extreme is glycine, in which as a side group it just have a small hydrogen. So what I told you before about the steric interference will be very small with glycine. So which one of these do you think belongs to glycine and which one of these belongs to proline? And this will determine if you understood the concept of phi and psi angles.
What is the acid?
So it's phi and psi, it doesn't matter which one. Okay.
This one would be glycine, the one in the middle.
This?
Yeah,
Why?
because it has a lot of oxygen formation.
Okay. And where is proline?
I think very
This? Okay, so you're correct, and if it's not clear to someone, then you can come ask me or ask in the forum. Don't be afraid of that. Like in general, So this is glycine and this is proline, and all the other amino acids are somewhere in the middle. Okay, and as you can see, a lot of the amino acids will not form U-turns, for example, at all. Okay, so and also glycine will not also tends to have a different pattern of orientation and will be less stable, like in beta sheets in a lot of the cases. So in the case of proline, you see that this is very restricted. Okay, so the amount of the flexibility of angles that this molecule can exist in is actually very restricted.
But it does seem that every amino acid can be formed into either alpha or beta.
Yeah, and you can see it also basically from here, because the actual amount, here it's just minor, like the formation. So it had to be like, it can also have been that this is just one amino acid.
It is for many amino acids.
Yeah.
The second one shows us like the specific
Yeah.
one.
Yeah, you're right. Okay. Question?
Just thinking about the shift within each cloud, I guess, depends on the other residues.
The shift within?
Like
You're talking about this or the other
like
one?
within every specific cloud, whatever. Yeah.
Yeah.
So within there,
It depends
on the other residues
of the other.
adjacent amino acids.
So in this plot, what they tried to do is like separate it. They actually did actually no amino acid exists freely. So they tried to separate each one of them. And yeah, in general, you can say that this is like a general, like the shift is mostly because of its own residue interacting with the other residues around it, or kind of average residues of the other residues that is around it. Okay. So U-turns, which are actually like less interesting than beta sheets and alpha helixes, are composed of 3 or 4 residues that are forming this sharp U-shaped turn. For example, here between this beta sheet that will be marked in this formation. So I showed you before that the alpha helix will be like a ribbon that's going like this, and a beta sheet will be normally in this orientation. And here you can actually distinguish like because I told you that a protein backbone has directionality, then we can also define the direction of the beta sheet. So the beta sheet can also, so if we define the directionality of the protein from N to C or from C to N, it's normally from C to N, remember? So if it starts here and ends here, then these two strands or this beta sheet will be considered as anti-parallel beta sheet. Okay? And if, for example, you had a structure that looked something like this, that did like a 3-dimensional loop and then returned here, okay? So these two will be in the same orientation, and you have a parallel beta sheet. Okay? So the parallel and anti-parallel beta sheet also has different types of hydrogen interaction between one another, and they are marked like in this way. If you see the two arrows that are contradicting each other, it's anti-parallel. And if you see them in the same direction, then it's parallel.
Yeah, what is what?
U-turn is just like these turns, okay, that are between, that are in sharp turns in the actual backbone of the protein. Okay, so in general, you can say that if you include like turns, beta sheets, and the alpha helixes, you more or less capture like most of the amino acids in the protein will be in either one of these formations. Okay?
Can you find the turn on its own without,
What do you mean?
like, you can see a beta sheet on its own and without
The beta sheet would have to have a turn, like inside,
Yeah.
in general.
But do you find only turns?
Only turns, like here.
Yes.
You can also have like general structure, you know. A lot of them are like, because of the 3D folding of the protein, you can have structures that are not defined into one of these categories. Okay? And also, a point that is important is that a lot of time we'll find glycine and proline, because of the opposite, like both of them will be found in U-turns a lot of times. Okay?
This
Yeah, because the thing is that here they define U-turn as especially like this angle, but a lot of times the definition of a U-turn can be, people also define it as this turn, not only this turn, you know. So I think in the last figure they regarded like U-turn as a very sharp bend, but a lot of time the point here is that proline, because of this kink that it forms inside the molecule, a lot of time introduces a turn of its own inside the backbone itself.
We can be only just like the connectivity between beta sheets and alpha helices and not exactly the same.
Yeah. What again?
Sorry, the U-turns can be defined also like only the connectivity between the beta sheets and the alpha helices and not exactly has to have a turn.
Well, let's just say that the U-turns are less defined than the other structures, okay? Because that's why I put them in the end. But so now we went over so the secondary structure in general is what we defined as alpha helix, beta sheet, and U-turn. And in the tertiary structure is actually the more complex three-dimensional arrangement of all the amino acids and residues. Okay, and remember that this is the last level that we're talking about the same molecule. Okay, so you can imagine that before we talked about the linear form of the peptide, which is the primary structure. Then secondary structure will be the alpha helix or beta turn, and then the tertiary structure is just the general folding of this whole molecule in space, okay? Which can be also subclassed, as we'll show in a minute, can be subclassified into larger components than alpha helix and beta sheets, but is a more like general form, okay? So in the level of the tertiary structure, and also what is in contrast to the secondary structure, the tertiary structure is probably stabilized by hydrophobic interactions and hydrophilic interactions between the side groups. So this will be the main thing that will determine, for example, if you have like two alpha helixes that are hydrophobic, then they will tend to cluster together in the center of the core center of the protein. Okay, and this interaction will be like a tertiary structure. So just to get you acquainted with how proteins look in general, or how in this case, so so this is an example of the Ras protein with a GDP molecule that is attached to it, so ignore that part. But so you can have different ways to how, or you will see also in articles, different ways to represent the protein in 3D. So the simplest form is just the backbone trace, just showing the backbone itself, without any added information regarding to helixes or beta sheets or stuff like that. The most complex way to show it is actually like ball and stick model, which actually shows each one of the molecules in this structure. So all the residues and except the hydrogen, I think that everything is depicted here. The ribbon model describes in a nice way like the ribbon formation of the alpha helixes and the beta sheets that you can see here. And my favorite formation is my favorite way of looking at it is the solvent accessible surface, which actually is created from a model that is trying to simulate if water can penetrate or molecules can penetrate inside the structure. And this actually shows you in a way like the steric interferences and steric interactions, and in the way that this protein actually looks. Okay, so this is how the environment sees the protein. It sees that as this bulb of electron clouds. That is, some of it has a positive charge, which is, for example, in blue here, and a negative charge, which is in red. But in general, you can see that it has volume. Okay, it's not any one of these orientations. So, in a sense, this is the closest that we can imagine to how the system or how the protein looks inside the cell. Okay, yeah. Okay. This is just negative charge. For example, in this protein, which is an interesting concept, a lot of times near the areas where you have another molecule that is bound, that is supposed to bind to this large structure, you will have a different charge or a dipole. So, for example, you see here that you have a lot of positive charges around the binding site of this molecule, and the red area around it. This actually helps to, for example, if this molecule is negative, then this will actually guide the molecule to the binding site. You can imagine that it will get repulsed and attracted to this area of the protein. Just a side. Because of electrostatic, for example, if this molecule. No, this is just my preference. But in a way, I'm saying it's a more real, more realistic way to represent it. Okay? Because when I try to imagine it like we see things. Okay? When you look at this, you just see the areas, you don't see the actual. Maybe it's not a good example, but in general, if you look at a protein, you will not see this structure. What you will see is what is important for interactions. For example, from this way of showing it, you can understand that maybe you can have a molecule that can cross readily, here, in this opening. Okay, from this illustration, but this is actually not the case. What actually is because you have too much steric interference around here, and also it doesn't show you all the backbones, and it's not showing you actually the whole molecule, because it's omitting several components for simplicity, just to show the general structure. And that's why I refer to this as more real life, because you also you see all the molecules that you really have, or all the atoms that you really have in the molecule, together with their electron cloud.
You might have to look at this from many different directions to see all the pockets and.
Yeah. Now, obviously, there are downfalls to any representation, and all of them are true. You just need to know what each one of them is showing. Don't look at this. The most important thing actually is not to say if I prefer one type and the other. It's just for you to understand the type of information that you're presented here. Okay? Not to get the idea that this protein really looks like this. Okay? That you have a lot of other components that are appearing here. Okay? Where's the thing? Okay. So, protein motifs are generally composed of several units of the basic secondary structures. Okay? So we were before in the secondary structure in the level of the alpha helix beta sheets, and now motifs are actually combinations of several of these motifs together. And why are we attributing to this level, and why are we talking about this level? Because because normally these motifs have specific functions. And for example, if you have a protein that have several types of motifs, usually it will mean that it has also several types of functions. So, like the alpha helix or the beta sheet, the structures were in a sense too small in order to say that, to attribute a specific function to them. We could just talk about the chemical properties of them and hydrophobicity and stuff like that. But we didn't talk about specific functions that they serve, and the reason for that is that normally, in order to talk about function, you need a larger structure. So these are examples of three typical protein motifs. This structure is the helix loops helix motif. So you have an alpha helix, then this loop over here, and then another alpha helix. And this motif is very typical when you find calcium binding, for example. So in a lot of different proteins, calcium binding. So in a lot of different proteins that bind calcium, you'll find a motif like that. And another important concept to understand is the consensus sequence, which will usually it will be around the actual binding site, and it will tell you what is the identity of the amino acids that you have to have in the order of them in order to form this motif. Okay? So for example, each amino acid, we didn't go over it, but each amino acid has a letter of its own. So you can have, so in the first position of the consensus site, you have to have either a D or an N, and then in the second position, it doesn't matter which amino acid you have, and then in the third one, you have to have either a D or N, then it doesn't matter, and then you have to have either D, N, or S. Okay? So in general, these consensus sequences are just empirically measured identities of different amino acids that people have found in these types of formations that can bind calcium. Okay? So this is not detached from the actual protein. Think about it that in proteins you can have like a whole huge protein and then a motif, a calcium binding motif. And similar to that, we also have other types of motifs. And these are just examples. We have thousands of different types of motifs that are known today. Another important type of motif is a zinc finger motif that associates with zinc, and together this type of alpha helix and then this connection area in a very small beta sheet together with a zinc molecule can bind DNA, for example. Okay? Did I add this? So RNA and proteins that bind DNA or RNA will usually have this zinc finger motif. Okay? And according to the fact that we find that, let's say we investigate a new protein and we see a calcium binding motif or a zinc finger motif, then we can understand something about its function in general. Another motif, which in this case doesn't have even to be inside the same protein, so that's why I don't like this example, but in general, it is this formation of a coiled motif in which you have two alpha helixes that have hydrophobic residues on one side of the alpha helix and hydrophilic residues on the other side. So here, because of the hydrophobic effect, these two will tend to aggregate together. And this form of aggregation will be found a lot of times in fibrous proteins that produce like fiber-like formations.
Why do they interact?
Because these residues are hydrophobic, and these residues over here are hydrophilic, like on the other side. Yeah.
The first two examples are one protein and the
Yeah. It not necessarily has to be one protein. Yeah.
in this specific, so in the
Here it has to be one protein, but here it doesn't have to be.
so in is that like a single protein can have a few kinds of
Motifs.
formations?
Yeah.
Doesn't have to be just one.
What do you mean?
And meant that a
It
protein
can-
is not obligated to be just with alpha helix.
No.
In the secondary.
It will not be, like the secondary structure is not, like, a protein will always be in its territory, I hate this word, tertiary structure, okay? It will always be in the tertiary structure, but the tertiary structure is composed of secondary structure that are mostly alpha helixes and beta sheets, okay? But once a protein, normally for each protein you have a specific orientation, a specific folding of that protein in space. And normally it will be just one. Okay? So one type of tertiary folding in space for a specific protein.
So this is just one.
But it can have several motifs.
Yeah.
Inside It can have several, like a lot of time RNA binding proteins have like a zinc finger motif and another motif that binds to a protein that this protein could be like a mediator between the RNA and other proteins, for example. Okay? So
Coil say, we said that it's hydrophilic and hydrophobic bonded together?
in this case, yeah. So you have hydrophobic residues on one side of the alpha helix and hydrophilic on the other side of the alpha.
Why do they bond? If
Bec-
some of them try to take water,
No, because
so they're not binding.
It's like the red and the blue are both hydrophilic. Or
hydrophobic. They're both hydrophobic. The red and the blue are hydrophobic, but they're just depicted here to show that they're belonging to different helixes. Okay, the red is from the right helix and the blue is from the left helix. Okay. Again, you can find it in one protein also. Like you can have this formation, but from here it's not necessarily one protein. Okay. So next time we're going to talk a little bit more about the structure, and we'll get very fast to the last the fourth structure. And so for next class, you just have to read the previous chapter that we talked about, that we finished like in the last class, and answer the question that we'll go over in the beginning of next class.
Just one question. Here, you could say that the motif is already the tertiary structure, or like a lot of motifs?
They're part of the tertiary. A motif is in the level of the tertiary structure. The only thing that is in the level of secondary is
the alpha and beta.
Yeah, alpha, trans, and beta. Okay. כן, כן.
לגבי הידרופובית והידרופילית. אז תכלס זה רק אומר שזה